Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Kerala SET Biotechnology question paper July 2023. I have solved the paper with explanations. This is part 1 of the question paper. Then if you are new to my channel, please don't forget to like, share and subscribe. I have also uploaded previous year question papers of Kerala SET Biotechnology. You can refer to my channel playlist for the same. Coming to the first question, lattice hypothesis explains. So lattice hypothesis, it is a hypothesis describing antigen-antibody reaction in terms of the buildup of large antigen-antibody aggregates or 3D lattices. So lattice hypothesis explain antigen-antibody reactions. Which among the following is an edible fungus? So Agaricus, Morchella, termitomites, all of the above are edible fungus. Shake flask culture is a batch culture. Somaclonal production is more possible during indirect organogenesis, because indirect organogenesis is via callus formation. So So higher chances of somaclonal variations can be observed in indirect organogenesis. So answer is option 5. Then cloning in which of the following vectors demands replica plating for screening recombinants? So out of the given options, PBR322 is the correct answer. Why? Because you can see the photograph. This is PBR322. It is cut. The restriction enzyme cut the tetracycline resistance region. A gene of interest is inserted in the tetracycline resistant gene region. So tetracycline becomes inactive. Only ampicillin resistance remains. Then plate onto media containing ampicillin after transforming the plasmid to the bacteria. Only those containing the recombinant plasmid will grow. Now you have to replica plate onto tetracycline containing mixture. So only those containing non-recombinants will grow on tetracycline containing mixture. So the answer here is option D, that is PBR322. Then next one. Which is the most probable reason for non-specificity of RAPD markers? It is low annealing temperature. Then glucokinase and lactate dehydrogenase are referred to as isozymes because, now what are isozymes? These enzymes differ in the amino acid sequence but catalyze the same chemical reaction. So in the options, option C, if you see, they catalyze the same reaction despite having different chemical structures. Different chemical structures means they differ in their amino acid sequence. So the answer is option C. What does the following equation represent? So this equation, it represents Eddie-Hofty plot equation for enzyme kinetics. Then ketone bodies which are produced in the liver are not utilized by hepatic cells. Why? Because the hepatic cells lack the necessary enzyme, that is beta-keto-acyl-CoA transferase. So they have not given beta-keto-acyl. They have only mentioned absence of CoA transferase. So that is the answer. Then chemiosmotic hypothesis of ATP synthesis in chloroplast is based on. So as per chemiosmotic hypothesis, ATP production is the outcome of the proton gradient that is established across the membrane of thylakoids. So the answer here is option C, that is proton gradient. Which of the following statements are not correct about the utilization of NADPH generated from the pentose phosphate pathway? So it is used for steroid synthesis. Yes. It is used for the regeneration of glutathione to its reduced state. Yes. It is used for the synthesis of fatty acid. Yes. It can be oxidized in the electron transport chain. ETC is electron transport chain to provide 38 ATPs. That is not correct. Then which of the following is not an essential amino acid? They have asked question from an essential and non-essential amino acid in the previous paper also, that was January 23. You can refer to my video for January 23 answers. So answer is, which is not an essential amino acid is serine. Then what are the products of urea cycle? So urea cycle is the process of conversion of toxic ammonia to less toxic urea. So if you see option A, they have told one molecule of ammonia is liberated. No. Option C also tells ammonia being liberated. Option D also gives ammonia being liberated. So that cannot be the answer. So option B is the answer. So even if you don't know urea cycle, you can answer this question if you know why urea cycle is being carried out. It is for the process of conversion of toxic ammonia to less toxic urea. So one molecule of urea, one molecule of AMP, two molecules of ADP and one molecule of fumaric acid. Which juice secreted by the organs in the alimentary canal plays a vital role in the digestion of fats? So it is bile juice and pancreatic juice. So answer is option. Then next one. Enzymes are used for the therapy and maintenance of various diseases. Applications of four major enzymes are mentioned below. Choose the correct application. So streptokinase facilitate wound healing. Streptokinase is used to dissolve blood clots that have been formed in the blood vessel. So that is not correct. Hyaluronidase promotes absorption of drug. That is correct. Collagenase. Collagenase, it is an enzyme that breaks down collagen in the damaged tissue and help healthy tissue to grow. That is, it facilitates wound healing. So what they have mentioned in the question, dissolves clot in myocardial infarction. So that is not correct. Chymotrypsin used for dissolving the ligaments of lens. That is also correct. So 2 and 4 are correct. Answer is option A. In an experiment, 0.1 ml of protein-free urea solution is allowed to react with 3 ml of a color reagent containing diacetyl monoxime and acid reagent in equal proportion. It is boiled at 100 degrees Celsius for 15 minutes and cooled. The concentration of the given sample will be. So here they have asked regarding Beer-Lambert law. So as per Beer-Lambert law, absorbance is directly proportional to concentration and path length. So here what they have asked, the concentration of the given sample will be directly proportional to the absorbance of light. That's correct. Directly proportional to the absence of light. No, indirectly proportional to the path length. It is directly proportional to the path length. Directly proportional to the temperature. It doesn't depend on temperature. So the answer here is option A, directly proportional to the absorbance of the light. Examples for predominant phosphogen or a macroergic compound that is present in vertebral, skeletal, muscle and brain. So phosphogen, they are also known as macroergic compounds, also known as high energy phosphate compounds, chiefly found in the muscular tissue in animals. Because muscle tissues can have sudden demands for more energy, and in these cases phosphogen supply the needed energy because they are high energy phosphates. So energy and they provide the required energy for the muscle that could not be immediately supplied by glycolysis or oxidative phosphorylation. So example of predominant phosphogen in the vertebrae, skeletal muscle and brain is creatinine phosphate. Answer is option C. Then next one. Application of radiotherapy to conquer cancer cells is done by bombarding the tumor cells with ionizing radiations. The basic mechanism involved in killing cancer cell using ionizing radiation is that radiation. So the major effect of ionizing radiation to any tissue or cell is direct killing of the cell by damaging the DNA. So whether it is cancer cell or normal cell, direct killing of the damage to the DNA molecule is taking place by ionizing radiation. So decreases nutritional level, no. Causes breaks in double-stranded DNA molecule, yes. Increases heat sensitivity in tumor cells, activates immune cells to prevent cancer. Which of the following is or are application of adsorption chromatography? Used for isolation of antibiotics, yes. Used to detect peptides, yes. Used to separate amino acids, all of the above. Answer is all of the above. Next question. Specify the features of photosystem 1. Maximum wavelength of excitement is 700 nanometers. That is correct. Uses chlorophyll A and B to absorb photons. No. Uses chlorophyll A to absorb photon energy. Yes. So both A and C are correct. Answer is option D. Which among the following is correct regarding non-polar molecules? So oxygen, nitrogen, carbon dioxide are non-polar molecules. +2 level chemistry. The reference electrode of a pH meter is a glass tube which is in contact with the mercury chloride block. It completes the circuit and is used to provide a stable zero voltage connection. Which solution is utilized to saturate the reference electrode? It is KCl. Answer is B. Then size exclusion chromatography, also described as gel permeation chromatography, is a technique that separates molecules depending on their size. Which among the following is true? Separation occurs via a specific binding interaction between mobile phase and stationary phase. It is only separation based on the size, so their specific binding interaction between mobile phase and stationary phase doesn't occur. Small molecules are eluted first. Yes. No. Small molecules are not eluted first. They possess more retention time in the between the inside. They enter into the pores of the gel. So they have more retention time in the column. So they elute only last. So B is also not correct. Large molecules stay in the porous beads in the column. They cannot enter into the porous beads of the column. So C is also not correct. Then smaller molecules possess more retention time. Yes, that is correct. Answer is option D. Then next one. A scientist wants to complete a ligation reaction in 5 minutes for an experiment to prove a criminal conspiracy and present the result on the same day to the criminal court of law. Okay. What will be his choice of ligation technology? Now you want the ligation reaction to happen in 5 minutes means T4 DNA ligase mediated ligation, E. coli DNA ligase mediated ligation take more than 5 minutes. So topocloning will be your choice because ligations by topoisomerase are extremely efficient and ligations can be performed in as little as 5 minutes. The next one, the maximum capacity of a lambda replacement vector is 24 kb. Answer is option C. The selectable marker which gene helps in SPI selection, it is red and gam mutations. So answer is option C. If A and B are the roots of the equation 2x square minus 14x plus 4 is equal to 0, then A square plus B square is. So a quadratic equation ax square plus bx plus c is equal to 0 with roots alpha and beta can be written as x square minus alpha plus alpha beta, where alpha plus beta is equal to minus b by a, alpha beta is equal to c by a. The given equation can be rewritten in the same format as x square minus 7x plus 2 is equal to 0. That is, you divide this equation by 2 or everywhere. So here alpha plus beta is equal to minus b by a, or minus 7 by 1, that is plus 7. Minus of minus 7 by 1. So it is plus 7. Alpha beta is c by a, so 2 by 1 is 2. Now a square, they have asked for a square plus b square. So a square plus b square is a plus b the whole square minus 2ab. So substitute the values. A plus b, that is alpha plus beta, is equal to plus 7, and 2 into ab, alpha beta is 2, so 2 into 2 4, that is 45. So the answer here is option C, that is 45. The equation whose roots are root 3 minus 5 and root 3 plus 5. We know that if m and n are the roots of a quadratic equation, ax square plus bx plus c is equal to 0, then the sum of the root is m plus n and the product of the root is mn, and the quadratic equation becomes x square minus m plus nx plus mn is equal to 0. Now substitute the value of m and n. Here m is root 3 minus 5 and n is root 3 plus 5. So you get 2 root 3x minus 22 is equal to 0. So the answer here is A, that is x square minus 2 root 3x minus 22 is equal to 0. Then next one. The targeted genome editing technology which make use of an RNA for site recognition is CRISPR, zinc finger nucleases, talons, megatails. So if you see this tabular column, these are megatails, ZFNs is zinc finger nuclease, this is talons, and this is CRISPR-Cas9. So mechanism of target DNA recognition for MNs, ZNFs and talons, it is protein DNA interaction. But for CRISPR-Cas, DNA RNA interaction via Watson-Crick base pairing is used. So here they have asked the question is, which targeted genome editing technology make use of RNA for site recognition? It is CRISPR-Cas9. So answer is option A. Then next one. The vector which are not using alpha complementation for recombinant screening. So out of these, which doesn't have lac is a DNA in their vector map, you have to see. So if you see the vector map of all these plasmids, except pcDNA, all the others have lac Z in their vector map. So pcDNA is the answer. Identify the thermostable polymerase with proofreading activity. So it is PFU. It is having proofreading activity. Answer is option C. Then how many hydrogen bonds are formed by the primer with the sequence 5' AGTCGGAATT 3' on annealing with the template? So how many hydrogen bonds will be formed? A will form 2 hydrogen bonds, G will form 3, T will form 2, C will form 3, G will again form 3, A will form 2 and T will form 2. If you add up all the hydrogen bonds, you will get it as 24. Answer is option B. Which among the following is codominant molecular marker? So codominant means both the alleles in the heterozygous sample will be detected. So it is RFLP is a codominant marker, whereas AFLP and RFPD are dominant markers. Then select the odd one among the following next generation sequencing platforms: Solid, pyrosequencing, Illumina, Heliscope. Of these, pyrosequencing, Illumina, Heliscope are based on sequencing by synthesis, whereas Solid, it is based on sequencing by ligation. So that is the odd one out. So the answer is option A. Then next one, apyrase. Apyrase is used in pyrosequencing of next generation DNA sequencing method. So A pyrase A3. So B and D can be eliminated. A and C we have to see. Then ligation is AB solid. I told you now sequencing by ligation is by AB solid. So B4. Lambda excision is LR clonase C1. So C is the answer. And PFU is high fidelity D. So the answer is option C. Then next one. Which among the following vectors are particular for animal cells? Cell lines. So it is animal cell lines. It is used to express proteins or commercial recombinant antibodies. So which vector are particular for animal cell lines will be pcDNA. The cis-acting region required for tDNA transfer. Vir protein, it is transacting site-specific endonucleases, no RO1 locus, no left and right borders. So left and right borders are cis-acting regions required for tDNA transfer. The microbe developed as a bioweapon, Clostridium bacillus anthracis variola virus, all of the above. Gene campaign initiative was started by Suman Sahai. Answer is option C. State whether the following statements about biosafety level 2 are correct or wrong. Herpes simplex virus is BL2 pathogen. BL2 is biosafety level 2 pathogen. Yes, that is correct. Daily decontamination is required in the facility. Yes, because it is a BL2 pathogen, you have to daily decontaminate it. So both 1 and 2 are correct. Answer is option A. Self-incompatibility in plants can be overcome by. So self-incompatibility, it is a mechanism that prevents self-fertilization and favors cross-pollination. So it can be overcome by embryo rescue method. If you see the options, other options, that is endosperm culture is for triploid production of triploids, ovule culture and other culture for haploid production. So self-incompatibility can be overcome by embryo rescue, and embryo rescue, it is normally used to obtain progeny from intraspecific hybridization techniques. Because intraspecific hybridization normally do not normally produce viable seeds. So answer is option A. Then next one. The easiest method to develop homozygous lines in plants. So endosperm culture for triploid production, embryo rescue I already told now. Then somatic hybridization, it is a novel technique that allows the fusion of two different plants to obtain a new hybrid plant with characteristics from both the plants. That is somatic hybridization. Double haploids. These are the haploid cells produced from pollen or egg cells can be induced or for chromosome doubling, and a double haploid cell is produced which can be grown into a double haploid plant. So the easiest way to develop homozygous line in plants is double haploids. Why? Because double haploids are developed from haploid plants which are homozygous. Haploid plants can be obtained from ovule culture or anther culture. Then next one. Macerozyme has dash activity. It has pectinase and hemicellulase activity. Then next one. An exclusive protein structure database. DDBJ is DNA data bank of Japan. EMBL is European Molecular Biology Laboratory. GenBank is an annotated collection of all publicly available DNA sequence, and PIR is Protein Information Resource. So out of these, all the three A, B and C are for nucleotides and only PIR is for protein. So exclusive protein structure database is PIR. Answer is option D. Then next one. Which among the following is not a multiple sequence alignment algorithm? Muscle MAFFT clustral. BLAST is not a multiple sequence alignment program. because it is mainly used to find the regions of local similarity between sequences. So it is not a multiple sequence alignment algorithm. So answer is option C. Then the standalone platform for DNA submission to GenBank. They have asked for the standalone platform. It is Sequin. Bank LT is a web-based one. So and standalone platform is Sequin. So answer is option A. Then next one. The software which is exclusive for maximum parsimony analysis is PAUP. PAUP. Answer is option C. Mr. Bayes, it performs Bayesian inference of phylogeny using a variant of Markov chain Monte Carlo. Then RAXML, it is randomized accelerated maximum likelihood, not maximum parsimony, it is maximum likelihood, is popular program for phylogenetic analysis of large data sets under maximum likelihood. Then MEGA is molecular evolutionary genetic analysis, is used for conducting statistical analysis of molecular evolution and for constructing phylogenetic trees. So they have asked specifically which is the one software which is exclusive for maximum parsimony analysis. It is POP. Answer is option C. The full form of UASB. It is UASB is upflow anaerobic sludge blanket. Answer is option A. It is this upflow anaerobic sludge blanket. It is a form of anaerobic digester that is used in wastewater treatment. Then next one, the major component in biogas. So biogas, it is formed by the decomposition of organic matter with the use by the action of bacteria in the absence of oxygen, that is under anaerobic condition. So the components of biogas are methane, carbon dioxide, nitrogen and hydrogen. So the answer here, they have asked for the major component, it is methane. Answer is option A. Then IPM stands for, it stands for Integrated Pest Management. Answer is option B. In B20, biodiesel 20 indicates the percentage of. So the blend percentage of biodiesel, it is indicated by B factor. So B100 represents pure biodiesel, whereas B20, it contains 20% biodiesel and the remainder being traditional petrol or diesel. So they have asked in B20, biodiesel 20 indicates the percentage of biodiesel component. Answer is option A. Then green manure plants belong to which family? So green manure are crops grown specifically for maintaining soil fertility and its structure, then supplying nitrogen and other nutrients, then preventing leaching of soluble nutrients from the soil, bringing crop nutrients up from lower soil profile, and smoothing weeds and preventing weed seedling growth. So out of these members, leguminosae are green manure plants. Why? Because they have symbiotic nitrogen fixing bacteria attached to their root nodules, and these plants enrich the soil with nitrogen. So answer is option C. Then rust and stirrers are not used in fungal bioreactors because it causes poor bulk mixing. Answer is option C. What is the height to diameter ratio of an ideal bioreactor? It is option A, that is 3:1. But in case of cell culture where gas flow rates are lower and mixing is gentle, the height to diameter ratio is 2:1. For ideal bioreactor, it is 3:1. Then, if log A by B plus log B by A is equal to log A plus B, then A plus B is equal to how much? So log A by B, log B by A can be written as log A by B into log B by A is equal to log A plus B. Log 1 is equal to log A plus B, or A plus B is equal to 1. So the answer is option A plus B is equal to 1. Then the commercial citric acid producing microbes, they are Aspergillus and Candida. Answer is option C. Then the full form of BASIC is Beginners All-Purpose Symbolic Construction Code. Instruction code. So answer is option A. Then next one. Which of the following is not a measure of central tendency? Mean, median, mode are measures of central tendency, whereas standard deviation it is measure of dispersion. So that is the answer. Answer is option B. Then next one. Oligonucleotides can be synthesized onto solid surface by. So oligonucleotide synthesis, it is the chemical synthesis of short fragments of DNA and RNA with defined sequences. So in solid phase synthesis, these oligonucleotides that are being assembled is covalently bound via its 3' hydroxy group to a solid support material and remain attached to it over the entire course of the chain assembly. Two most important solid phase materials are pore glass and macroporous polystyrene. Then they have not given laser pore glass and macroporous polystyrene, but they have given photolithography. Photolithography is a powerful technique for the synthesis of DNA oligonucleotides on glass slides. So it is a solid surface method. So answer is option A. What is laser ablation? Laser ablation is used in science to destroy nerves and other tissues to study their function. That is called as laser ablation.